Hello, good afternoon. I'm Gisela Terwindt. I'm a neurologist and biologist at the Leiden University Medical Centre. I work with a big team on genetics of migraine. I'm especially interested in neurogenetic. When I started, I wasn't that interested in migraine, but I just came into the field by meeting Professor Ferrari. What I will do today is to discuss with you the genetic architecture of migraine, but especially the lessons learned so far and for the future. These are my disclosures. But let's start. So how can genetics set the path? Well, first of all, I think genetics can be very important for diagnosis and classification based on disease biology, which is not the case currently in our ICD classification. The only genetics that is involved in our classification is the distinction between hemiplegic migraine type 1, 2, and 3, and I will come to that later. But I think in the future it might be that genetics are extremely important also for classification systems. Then the prediction of prognosis. I think genetics can also play an important role in quantification, the prediction of which persons are more prone to bonify and which persons are not. And also to study comorbidities and complications, for instance, the stroke risk that is among migraine patients. And last but not least, I think the selection and prediction of response to treatment, and that's called precision medicine, might be very important in the future, and genetics can help with that as well. Migraine is a hereditary disease, meaning that in many patients with migraine there is familial clustering, and this is especially the case for those with migraine with aura more than for those with migraine without aura. And there is a special form of migraine with aura that you might be familiar with. It's called familial hemiplegic migraine, and it has an autosomal dominant inheritance, and I will come to that later if that's the case for all, but for most patients that means that when a parent suffers from the disease, the offspring is likely to suffer from hemiplegic migraine in 50% of cases. However, there are also sporadic hemiplegic migraine patients, meaning that only one person in the family is affected. And this could be that it's a de novo mutation, so a new mutation is occurring, and it's likely that a familial hemiplegic migraine will develop when new generations are born, but sometimes no mutation is found in these sporadic cases, and I will come to that as well. So what's hemiplegic migraine? It's a clinical diagnosis, meaning that people do not only suffer from headache, but also from aura, and with these aura symptoms they need to suffer also from motor symptoms, meaning that they have a hemiparesis or even a hemiparalysis of one side of the body. And I often see that neurologists are confused, and they send to me patients and asking whether it's hemiplegic migraine patients, and patients describe sensory tingling feelings of one side of the body, and that's not hemiplegic migraine. What do we know about the genetics so far? So the first gene that we discovered was in 1996. It was during my PhD time, and it was the CACNA1A gene, calcium channel gene. That is involved, and we call these families and patients FHM1 according to the classification. The second gene that was discovered is the ATP1A2 gene, an ATPase gene, and the third gene that you might recognize this from epilepsy is the SC1A gene. It's a sodium channel gene. So 3 genes have been shown to be implicated in hemiplegic migraine, but still some patients do not have this mutation, and I'll come to that. But when you identify a pathogenic mutation in one of these 3 genes, there is a confirmation of clinical diagnosis. So what about those who do not have a mutation in one of these genes? Are there novel genes to be discovered, or are they already discovered? And what about the phenotype? Maybe they have displayed another phenotype. So what do we do with this patient with no pathogenic mutation? So we study in Leiden, at the Leiden Medical Centre. We took our database, and I think we have the largest database on hemiplegic migraine worldwide. And we had 68 families, 268 cases in these families, and also sporadic hemolytic migraine cases, those were 33. And we looked whether we could identify mutation. And in 208 of these patients, we identified a mutation in one of these 3 known genes. But we also saw that in 73 of cases, we couldn't find a mutation in the known genes. And also we identified some unclassified variants, of which you can say that it's probably not a variant that is implicated in the disorder. So we took unique cases from these patients that were not shown to have no mutation, and we did whole exome sequencing in these 47 unique families and patients. And no novel hemolytic migraine genes were discovered. There's also another gene that is in the literature, and that's called PRT2. And maybe you've heard of it. It has associated phenotypes, being benign familial infantile seizures, for instance, but also paroxysmal kinesiogenic dyskinesia, and also the ECCA syndrome. So all convulsions starting at a young age. Be aware that the vast majority of these PRT2 mutation carriers do not have hemolytic migraine. This is an example of a family we had. And I have to confess that we published this family. And this family showed both hemolytic migraine and benign infantile convulsions. And we first published this family as proof that ATP1A2 could not only cause hemolytic migraine but also convulsions. And in the end, we looked further into this family, and it was shown that also the PRT2 mutations occur in this family. So there's a combination of 2 genes involved in this family. So PRT2 mutation carriers with hemolytic migraine nearly always also suffer from other PRT2 related phenotypes, as shown in this family. Furthermore, several PRT2 mutation carriers with hemolytic migraine also have a pathogenic mutation in CAT1 or ATP1-2. So we thought PRT2 itself will not cause hemolytic migraine. It's an add-on gene that may be involved. What's the role of PRT2 in hemolytic migraine? Of course, it affects neuronal excitability, but we sincerely think it's a genetic cofactor causing hemolytic migraine, but not being the 4th FHM gene. So when you find a pathogenic mutation in PRT2, it's not a definite confirmation of clinical diagnosis. And I see a lot of new literature, and also recently a new publication was also published, and it was also reviewed by headache experts in Lancet Neurology, indicating that PRT2 would be the 4th hemolytic migraine gene, but it is not. It is a comorbid factor playing a role, making it more likely that people may develop both convulsions and hemolytic migraine attacks, but it's not the same. It doesn't have the same strength as the known 3 other genes. That's very important. And why is this also important? Because it also indicates, and I will come to that later, that it might be that in this unknown group, where not the main 3 genes are involved, other phenotypes may occur, but also other genetic factors play a role. Not one gene implicated in these families or patients, but multiple genes leading to this hemolytic migraine phenotype. So I think this slide is extremely useful when you're in clinical practice. So I use it also all the time. So when a patient comes to your clinic, you can actually predict whether it's likely that one of the 3 known genes will be implicated in that patient. So based on the ictal clinical features, when attacks are triggered by mild head trauma, or when hemiparesis is very severe, including the facial paresis or hemiplegia, or there are 1 or more brainstem auras features, then there is an increased chance of finding a mutation. Also, looking at ictal diagnostic features, if you have CSF pleocytosis during attacks, or edema on one hemisphere on ictal brain MRI, it's more likely that you will find a mutation in one of the known genes. When the age at onset is below 10 years, and there are 2 or more family members with hemolytic migraine, it's more likely that you will find a mutation. And when there are chronic features like mental retardation or progressive chronic ataxia, it's also likely. If these features are not the case, it's much more less likely that you will find mutation, and you can even question yourself whether you should do genetic testing in those patients. So this is a proposal that we have. We think that on one spectrum you have migraine without aura, and you have migraine with aura, and I come further on the genetics of those. And on the other hand, you have this severe hemolytic migraine 1 through 3, where there is extensive motor aura, but also other features as indicated here with brainstem auras, head trauma triggering, brain edema, ataxia, mental retardation, fever, confusion. In these patients, it will likely that one of the 3 genes are implicated. But in between, there seems to be a MA plus phenotype, where patients have more than what we would say the common aura phenomena, with some motor aura symptoms, but not as severe as the FHM1, 2, 3. And in this MA plus phenotype, it might well be that PRT2 is a cofactor involved, but not the main causing gene. So when you have, also what we saw is that when you have this MA plus phenotype, there are less affected relatives, so the inheritance is much more unclear. So a pathogenic mutation in one of the known genes can confirm a clinical diagnosis of hemolytic migraine, and a mutation in PRT2 cannot. Furthermore, most patients with hemolytic migraine without a mutation have a more mild phenotype, more akin to that of the common non-hemolytic migraine patients. And motor aura may not always be monogenetic and thus not necessarily inherited by 50% of offspring. And that's also important for your clinical practice. If you have a patient with motor aura, you shouldn't say it's likely that you have an autosomal dominant inheritance and it's likely that your children will have a chance of 50% to inherit the disorder. No, it's probably less than 50%. So this is very useful for the clinical practice of neurologists, I think. And furthermore, at least, a major fourth autosomal dominant gene for hemolytic migraine remains to be identified, but I would say is rather unlikely. So let's move on to the more common types of migraine, because when hemolytic migraine itself, or the MA plus, is already quite difficult, what about the more common types of migraine with and without aura? Well, we know they have a complex inheritance, meaning that multiple factors are involved, multiple genetic factors, but also environmental factors. And to study these multiple genetic factors, we do genome-wide association studies, where you use SNPs. They are just kind of flags within the DNA, and you try to identify the flags and correlate these flags and the color of these flags to whether a person is a patient, a case, or a control, a non-migraine person. And the question is, can we use the GWAS data already for diagnosis-based disease biology? So this is the last publication that has been done by the International Headache Genetics Consortium, and many researchers are involved in this. We as the Leiden group, but also the Danish group is very much involved. So I think it's a great consortium and we do a lot of work together, and I think the only way to come to this kind of papers is by working together, because as you can see, more than 1,000 migraine patients were included, and also more than 7,000 controls were included in these genome-wide association studies. And so far we identified 103 risk loci for migraine. Interestingly, some of these risk variants seem to be specific for migraine with aura, where others seem more specific for migraine without aura. But still, we should be aware that in these large cohorts, the phenotyping of the aura is not always that optimal. So I think we need confirmation for this kind of findings. But overall, variants were enriched for both vascular and central nervous system, supporting a neurovascular mechanism. So we think we had the discussion yesterday: is migraine a neurological, is it a neuronal disorder? Is it a glial cell disorder, it's the vascular disorder, and I think based on the genetics, you could say there are implications for both mechanisms, so that's why we call it a neurovascular disease. Interestingly, if you look at the GWAS data, you can also find loci that we now know are important for treatment. So if you look back, we now know that CGRP is extremely important. And if you look back now to the GWAS data, you find indeed genes implicated in the CGRP pathway. So as a proof of principle, you see that GWAS cells identify those targets that are important. And it's also true for serotonin 1F receptor, and we know it's useful in clinical practice for treatment. And when you look in the GWAS data, you see those SNPs indicating those genes, pathways that are implicated in serotonin 1F receptor. And this is just a proof of principle, because what we need to do, of course, is to identify targets that we're not aware of. And that's quite more difficult, because we have only these SNPs, these flags. These are not the genes itself. So we have to look around these SNPs, which genes are implicated. We have to do pathway analysis, and then we can hopefully identify new treatment targets. And what I, for instance, are hoping for is new treatment targets, especially for those with migraine with aura. Because my impression is that it might be that CGRP is not the main initiator in migraine with aura patients, and therefore it might be that migraine with aura patients who suffer from multiple aura attacks, not one aura and 90% of them without aura, but those who have in 100% migraine with aura attacks or even hemiplegic migraine attacks, we don't have adequate treatment for them yet. So I hope that GWAS studies can help to identify these new targets. So I'm talking about the GWAS data, but we're now entering what I call the post-GWAS era, where we are going to look, what are we going to do with this GWAS data? So we can look with Mendelian randomization, and I will come to that, looking at the causal effects. I tried to explain already to you that I think we need to identify new pathways to develop new drug treatments for patients. I will come to polygenetic risk scores, where you use GWAS data to predict the involvement of genetics in your patients. But in the end, we want to come to precision medicine, where we can identify those patients for that treatments that we think are optimal for them. So personalized medicine. So let's start with the polygenetic risk scores. So when you have this GWAS data and you have all these SNPs, you can actually calculate what's the genetic risk for your patients. That's called polygenetic risk scores. And this is an example of that. What you can see here is that when you can compare Finnish controls, that's in the upper side in red, and you compare them with population-based, this point is really tricky. Population-based cases, you see that migraine cases have a higher polygenic risk score. Yeah, that's what's called. They have more genetic factors involved for their migraine. But when you compare those who have no family history with those with any family history, you see that it increases more. Oh, and the pointer, this is, yeah. And if you compare families with migraine with or without aura, with those with families with typical aura and those with hemiplegic migraine, you can see that the polygenetic risk scores increases, meaning that in these families with aura, hemiplegic migraine, more genes are implicated than in those with migraine without aura. Is this helpful? The first statement would be there is a significant contribution of common polygenic variation to the familiar aggregation of migraine. Okay, nice conclusion. So let's start with the bottom of the slide. So if you calculate polygenic risk scores, you can actually predict whether there's a stronger family history of migraine. You can predict that there will be a lower age onset. You can predict that it's more likely that patients will suffer from migraine with aura. but you cannot predict yet whether there's efficacy of migraine-specific drugs. In clinical practice, we just ask our patients, do your parents suffer from migraine? And that's the top. So if there's a family history of migraine, and when a patient comes to my clinic, I ask, do you have migraine in your family? Does your father suffer from migraine? Does your mother suffer from migraine? And if they say yes, then I know that it's likely that they have a lower age of onset, that they have a higher migraine frequency, especially in males. And I think that's because in females hormonal influences are extremely important. They will likely have a higher number of medication days, and it's more likely that they will suffer from migraine withdrawal. So currently, for my clinical practice, I don't need these polygenic risk scores. I just ask about the family history. So, and also this is indicated in this slide, if you would calculate, if you would do a SNP analysis in each of your patients, it will only explain in a population-based case 1.6% of the variation, and in a familial case only 3.5%. So for now, there's no role of determination of GWAS SNPs in the clinical diagnosis of migraine. So for that purpose, it's not useful. But for other purposes, it will be more useful. But for the clinical diagnosis, it's not useful at this moment. Where is it useful for? For instance, when you are looking and you want to investigate the relationship between migraine and its comorbidities, and also the complications, then we can use the GWAS studies. And there are great examples of that, and I will show you that. One of the examples is the combination of psychiatric disorders and neurological disorders. And yesterday, we had a psychiatrist who talked with us about GWAS and also about the overlap between psychiatric disorders, and I was wondering, well, is their classification not correct? Are we better in classification? And that's why they have this overlap. So what we did in this brainstorm consortium, we looked at the GWAS data of neurological disorders and for the psychiatric disorders. And for neurological disorders, there was an indestructible sharing of common variant risk. So there was no genetic overlap between migraine, epilepsy, stroke, MS, Parkinson's disease, Alzheimer's disease. This could be due to several factors. Either there's no relationship between migraine and Alzheimer's disease on a pathophysiological level, that would be a good explanation. Could be that we don't have enough SNPs identified yet to find this overlap. Or it could be that we are very good in classification of neurological diseases, and that's why there's no overlap between those disorders. For psychiatric disorders, there was a substantial sharing of common variant risk. That was also explained yesterday. This could be because there is no good classification system, would be one example. Another example would be there are more embedded GWAS data for the psychiatric disorders, and that's why they find this overlap. Or it could be, I think that's the most interesting part, there is indeed a genetic overlap between these psychiatric disorders. And now comes the interesting parts. There was a huge overlap between the GWAS of migraine and major depressive disorder and anxiety disorders. So it seems that migraine is genetically more related to psychiatric disorders than to neurological disorders. And I always try to translate this to clinical practice. We know that migraine patients are more likely to suffer from depression and anxiety. We also know that when you have a patient who's more likely to, that is more likely to suffer from depression and anxiety, they're more likely to have medication overuse and that they will chronify. So depression and anxiety is a risk factor for chronification for our patients. We also know that, we did a study ourselves, that when patients get a migraine attack, they will be more depressed during the day of a migraine. Just that's very logical for me. You're in pain, you have to go to bed, so you're depressed. Of course you're depressed. But before the attack, they're less depressed, and after the attack, they're also less depressed. So there is a peak during the attack, but at the same time, there is a comorbidity making people more prone to chronify. So many people ask to me, so it's my fault that I have this medication overuse and chronification? And then I say to them, no, you are more vulnerable because you have this genetic predisposition. This genetic predisposition makes you more likely to suffer from both from migraine and both for depression. And we have to monitor that. And in clinical practice, it means that every patient that comes to our clinic is screened for depression and anxiety. And if there's depression and anxiety, we just discuss it with our patient. Is the current problem? Should we have some more awareness to this? Should we have more? We cannot provide this guidance for depression and anxiety at our hospital because it's a national hospital. People come from all around the Netherlands. But we say to them, go to your GP, because we think it's an important factor that should be taken attention to, and maybe you should go to a psychologist, not because we think that you're crazy of your migraine, but because we know it's an important risk factor. So this is how I take this knowledge of genetics in clinical practice, into my clinical practice, and use it in my clinical practice. One other thing that you can do with GWAS studies is look at causal relationships. So, and that's called Mendelian Randomization. I'm not sure if you're familiar with it, but I will try to explain to you. So, for instance, you have an exposure. You want to study whether high blood pressure is causal for migraine. So your patient comes to you and she said, I have high blood pressure, I have migraine. Is this causal? Is this high blood pressure causing migraine? I think it is. This patient said. And then we can study this kind of relationship by doing Mendelian randomization. So with this, exposure is high blood pressure. And you take the GWAS data for high blood pressure. So the assumption is that you select those SNPs, those flags, that were associated with high blood pressure. The other assumption is that these SNPs are not associated with other confounds. For instance, high cholesterol or other cardiovascular risk factors that might be implicated also in migraine. So these SNPs are only involved for high blood pressure. And these SNPs, these flags, should also not be involved in migraine itself. So they should also need to be independent of the outcome. Because sometimes you have actually an overlap between the SNPs for the exposure, high blood pressure, and the migraine. For instance, if you would look at depression as causal factor for migraine, we now already know that the SNPs that are involved in depression are also involved in migraine. And that's what we call pleiotropy. I can hardly pronounce it. Horizontal pleiotropy. So meaning that these genetic factors are implicated in both the exposure and the disease. And with that, you cannot do the cause-effect Mendelian randomization. So there are many complicated statistical analyses that you can do before you can study this cause-effect relationship. So when you look into the literature, there are many Mendelian randomization publications these days. But take a good look and pick those out who are really good. So I took one as an example. It's from Dale Nieholt. He's also in our International Genetics Consortium, and he's a very good genetic statistician. So this is an example of how you can do that. So they looked at 9 clinical chemistry tests. 5 were related to CVD markers, for instance, hypercholesterolemia or something like that, iron markers and liver markers. So they just had some clinical chemistry tests. And they took the GWAS data, the summary GWAS data from migraine, that's the disease, the GWAS from the clinical chemistry test, that's the exposure, and also the GWAS from the medical relevant disorders linked to this chemistry test, so cardiovascular disease, iron deficiency, and liver disease. And then they looked whether there's a causal relationship. And I think the most important conclusion of them was that there was already an immense genetic overlap. So there were many shared genetic effects for the risk of migraine in some of these markers. And these genetic factors also influence these markers, are shared across migraine, cardiovascular disease, iron deficiency, but not liver diseases. So in the end, they concluded that most importantly, there was this pleiotropy, horizontal pleiotropy. So there was a shared, these GWAS SNPs, they were shared among these markers and diseases. And with with that, you cannot look for the causal effect. So it's very unlikely that this clinical chemical test and medical traits causally affect migraine. So to translate it, if you have a patient, a migraine patient with a high cholesterol, and the question is this high cholesterol causing my migraine? This kind of analysis proves that the high cholesterol itself is not causing a migraine. But it might be that there's a genetic overlap between causing the high cholesterol and causing the migraine. Yeah, is it clear for everyone? Because it took me a while to understand this. So I hope I explained it. I think this is the new approach that will become coming the upcoming years, and it's very very nice to look into it because it's very important also for clinical practice. So this is my last slide to wrap up. So what about genetics of migraine in the post-GWAS era? So GWAS are extremely helpful to identify pathophysiological pathways and potential drug targets. But we need to put effort in it. So we shouldn't be happy with this new publication with 123 load sites. It's great, but we have to go into the depth, look at pathways that are implicated. Furthermore, the assessing all these SNPs, these flags in our patients, it's not helpful for clinical practice at this moment. It's not helpful at all. So we shouldn't do that. Just ask about family occurrence. And then you will know that it's more likely that if a father or mother is affected, that your patient will have a younger age of onset, have more medication use, will be migraine with aura patients, etc. So just use that in your clinical practice. And I explained to you the Mendelian Randomization method because I think it's extremely important and exciting to look for causal relationships between exposures and migraine. And this will help us in the end to come to precision medicine or personalized medicine where we can identify those patients who will be likely to respond to a certain treatment that we provide to them. And I would like to end my presentation with that and also make some advertisement for another symposium that we're organizing in Leiden. It's called the Leiden Headache Center into a New Era. If you want to look, you can look it up, Google it up, and you can also join us in June. Thank you for your attention. Are we having questions? Short question.
5 minutes. Okay, only 5 minutes. So I don't
only urgent questions.
Yeah.
Maybe you can just speak out loud and I will repeat it.
I can, yeah.
Maybe you can ask Kashmar to help you.
No, it doesn't work. But you can hear me maybe. Yes?
Yeah, if you speak out very loud, we can
Thank
hear.
you for a very nice presentation. My question is about the FHM migraine types and different gene where you detected mutation. Are we speaking about the same mutation in the genes or different mutations? If you got my question, is there several types of mutation, or it's the same mutation you detected on all these genes?
Yeah.
This is the first question.
Okay, I will answer your first question first, and I will repeat it for those who didn't hear it. The question is, there are 3 known FHM genes, CACNA1A, ATP1A, and SCN1A, and is there only one mutation in those genes, in each of these genes implicated in migraine? And the answer is no. So in CACNA1A there are multiple genes at different positions in the gene itself that may affect a person and may lead to hemolytic migraine. That's different from other. There are some other disorders where only one mutation at a certain point will cause the disease. But for migraine, it's much more complicated. Even for FHM1, there are multiple places on the CACNA1A gene that may be affected and may lead to hemolytic migraine.
Okay, and this gene we are speaking about, it's not a control element, DNA element, that controls other genes, maybe.
No, so, yeah, okay. So to repeat the question, in this, I think FHM 1 and 2 and 3 are actually quite classical autosomal dominant disorders, meaning that if you have a mutation in the gene, it will affect you, and we know the common pathway for all these genes is in the end there is an increased glutamate and potassium in the synaptic cleft, and this leads to an increased risk for cortical spreading depolarization, and that's the underlying mechanisms for the aura, so that leads to the impinging migraine attack. So it's, for those disorders, it's totally clear what the pathway is.
Thank you.
Yeah?
Okay, one last question from Anders.
It's working now. Thank you for a very nice presentation. I think that this concept of a migraine with aura plus subgroup is very interesting. Do you routinely screen this patient group for the known FHM genes and other genes at your department?
Yeah. So I understand your question. When we started, we only investigate our patients for FHM 1, 2, and 3, and when no mutation was found, we stopped. There was even a time where we said, why would we screen at all? What's the use of it for clinical practice? Are we going to treat our patient differently? No. So we don't do exome sequencing for patients who do not have a mutation, because it won't help us. We will find all kinds of variants, unknown variants in clinical practice that are not useful. For research purposes, it's interesting, of course, because you want to look for new genes. For clinical practices, it's no use at all. And what I now see in the Netherlands and maybe all over the world is that when people encounter even quite common migraine with aura families, and they start screening, they start to screen also for PRT2, and they find a PRT2 mutation, and sometimes even clinical geneticists write a letter stating that PRT2 is causing migraine with aura in these families, and I think that's totally wrong. It might be that PRT2 is implicated as a cofactor, but it will never be the one and only gene and mutation that's implicated in those families. So it's wrong to say to those families, you have a mutation that proves that this explains your disease. That's totally wrong. Yeah.
Thank you, Professor.
Yeah.
Very excellent presentation.